Hello everyone, welcome to the next video in our physiology video lecture series. This is the second part of our lecture on heart. This particular lecture will cover the regulation and control systems of the heart, which control the rate of the heartbeat as well as the strength of contraction of the heart. Further, we'll look into how exactly the rhythmicity of the heart is generated, what exactly generates the heartbeat in the first place, and how this action potential that is generated in the heart itself is transmitted to the other parts of the heart so that it can beat as a syncytium, as we discussed in the last video lecture. To start off, let us look at the regulation of heart pumping. During rest, a person's heart pumps about 4 to 6 liters of blood each minute. But during exercise, this volume increases dramatically to 6 to 7 times the normal resting volume. So, what exactly occurs that changes the heart pumping rate so much? This regulation is done via two methods. First is the intrinsic cardiac regulation, or the regulation that is done by the heart by itself, which is also known as the Frank-Starling mechanism. The other is the control of heart rate and strength by the autonomic nervous system. So the nervous system, as we know, is divided into two parts: the autonomic nervous system and the voluntary nervous system. Autonomic nervous system is not under our direct control, and this is the part of the nervous system that controls the or regulates the heart pumping. So first, let us look at the intrinsic regulation or the Frank-Starling mechanism. Under most conditions, the amount of blood pumped by the heart each minute is determined almost entirely by the rate of blood flow into the heart from the veins, which is called the venous return. So, in normal conditions, the rate or the volume of the blood that the heart will pump depends directly on the amount of blood that is coming into the heart. This blood that is coming into the heart is known as the venous return. The heart, in turn, automatically pumps this incoming blood into the arteries so that it can flow around the circuit once again. This intrinsic ability of the heart to adapt to increasing volumes in the flowing blood is known as the Frank-Starling mechanism of the heart. What this means is that if we increase the venous return to the heart itself, so more blood is coming into the heart, then when the heart pumps, more blood will be pumped out of the heart, which is obviously instinctive, and this is known as the Frank-Starling mechanism. So more blood coming into the heart means more gets pumped out. But what is the cause of the Frank-Starling mechanism? As greater volume of the blood flows into the ventricles, the cardiac muscle stretches to a greater length. So you can consider this situation as a balloon. So more air you push into the balloon, the greater it stretches. Similarly, the more blood or the more venous return, the amount of stretching of the heart muscles increases. This in turn causes the muscles to contract with greater strength as actin myosin filaments are brought to more optimal degree of overlap for force generation. So once the heart muscles get stretched to a longer distance, you can consider this situation as that of a loaded spring. And thus, when the heart muscles contract, they can contract by a larger degree and thus generate greater force, pumping a larger amount of blood out of the heart. This extra force is what allows the ventricles to pump extra blood into the arteries and is the reason for the Frank-Starling mechanism. So we move on to ventricular function curves. So this increase in blood outflow, how will we exactly quantitate this increase in blood outflow from the heart? This is done using ventricular function curves. The functional ability of the ventricles is best shown by the ventricular function curves. The ventricular function curves are of various types, one of which is shown here, which is the ventricular volume output curve. which is a plot of ventricular output, so the blood that is coming out of the ventricles, with respect to arterial pressure. And as we can see in this particular plot, as arterial pressure increases, the ventricular output also increases dramatically with it. So there is almost a linear increase in ventricular output with respect to arterial pressure, after which it saturates. So these curves demonstrate for both the right ventricle and the left ventricle that the Frank-Starling mechanism of the heart works at a higher arterial pressure. That is, at higher arterial pressure, more blood is getting pumped into the ventricles, and thus, as more blood gets pumped into the ventricles are filled with higher volume of blood, and strength of muscle contraction increases. The heart then pumps higher quantities of blood out of the arteries. So, what does the arterial pressure mean? Arterial pressure means that the force with which the arteries are contracting, and can be taken as a proxy for the amount of blood that the arteries pump back into the ventricles. As arterial pressure increases, this means that the amount of blood getting into the ventricles increase, and as such, and as the amount of blood that gets into the ventricle increase, we see there is an increase in the ventricular output, or the pumping of the blood out of the heart. This shows us directly that the Frank-Starling mechanism is working in a normal heart. That is, with more blood getting pumped into the ventricle, more blood gets pumped out. So this was the ventricular function curves. Next, we move on to the control of heart by the nervous system. So after dealing with the intrinsic control, we move on to control of the heart by the nervous system. So as I said, the heart beat is controlled by the autonomic nervous system. But the autonomic nervous system is also further divided into sympathetic and the parasympathetic nervous system. So, what are those? The sympathetic nervous system is the nervous system which excites various parts of the body which respond to the flight or fight response. That is, the stimulation by sympathetic nerves prepares the body for a flight or fright or excitatory response, that is, movement of some kind. So this can include dilation of the pupils, more adrenaline getting released, increase in heartbeat. All these are examples of stimulation by the sympathetic nervous system. Parasympathetic nervous system, on the other hand, is the part of the nervous systems which when stimulated, produces a resting effect in our heart. So, resting effect in our body. So, this parasympathetic nervous system stimulates the digestion and the resting of our system. So, it will start preparing the stomach to digest food as well as lower the heart. So, both of these, sympathetic and parasympathetic nervous system, affect the heart and control it. So, as I said, the sympathetic nerves act during the flight or fright response of the body, while parasympathetic nerves are active during the resting phase of the body. Thus, sympathetic nerves can increase the amount of blood pumped each minute by 100%, like it doubles the amount of blood that is pumped. Whereas vagal or parasympathetic nerve stimulation decreases the output to as low as zero. So, as I said, sympathetic nerves increase the amount of blood pump, and parasympathetic decreases the amount of blood pump out of the heart. They also affect the heart rate, and we will look into how they do it. But first, I would like to point out that we are able to see various nerves from the sympathetic nervous system innervating the heart, but only the vagus nerves that that innervate the atrial part of the heart. So the vagus nerve represents the parasympathetic nervous system and innervate mostly the atrial part of the heart. So we move on to discuss in detail about the control of the heart by the nervous system, and look into details about the characteristic of control by the sympathetic nerve. Sympathetic nerves innervate the heart at various locations, as we just saw. The strong sympathetic nerve stimulation can increase heart rate from 70 beats per minute to about 180 to 200. and rarely to 250 beats per minute. That is a extreme state of excitation. Further, sympathetic stimulation can increase the maximum cardiac output to 2 to 3 times normal. Inhibition of sympathetic nerves to the heart can decrease cardiac pumping to a moderate extent. So, even in normal conditions, the sympathetic nerves are already stimulating the heart, which maintain the normal heart rate. But inhibition of sympathetic nervous system completely also decreases the cardiac pumping. Finally, when activity of the sympathetic nervous system is depressed below normal, this decreases both the heart rate as well as strength of ventricular muscle contraction. So not only is the heart rate affected, but the strength of the ventricular muscle contraction is also affected. Next, we move on to excitation by parasympathetic nervous system. The parasympathetic nerves stimulate the heart via the vagus nerve, mainly to the atria. So the vagus nerve only innervate the atria, or mainly innervate the atria. Strong parasympathetic stimulation can slow down or even stop the heartbeat. This can occur, but if even in the case of strong parasympathetic stimulation which stops the heartbeat, the heart can escape and start beating again, which is known, which is a phenomenon that we will discuss towards the end of the slides. The parasympathetic nervous stimulation can reduce the heartbeat to 20 to 30 beats per minute and decrease the strength of heart muscle contraction by 20 to 30%. The innervation of the atria by the vagal nerve dictates that the vagal stimulation is mainly attributing to decrease in rate of contraction and not the strength of cardiac contraction. This is an important point, as we have seen. The strength of the cardiac contraction depends mainly on the ventricles, as those are the major pumps that pump blood into the body. So they are the ones that dictate the output of the heart. And the atria mainly, or the parts of the atria mainly dictate the rhythm or the rate of heartbeat. And thus, as the parasympathetic nervous system via the vagal nerve mainly innervates the atria, the parasympathetic nervous system controls the rate of heartbeat much more than the strength of cardiac contraction. Next, we have a diagram which represents the effect of cardiac output due to various nervous input. In the blue line, we have normal sympathetic stimulation. When the sympathetic stimulation is increased, we see the red line, which is way higher than normal and gives us around 24 liter per minute cardiac output. But in case normal sympathetic stimulation is completely removed, we see a decrease in the cardiac output to the green line. As I said, even in normal conditions, sympathetic stimulation is necessary. And when further parasympathetic stimulation is given, the cardiac output further decreases to about 8 liters of blood per minute. Next, we go into the effect of various ions as well as temperature on the functioning of heart. So the effect of, we first look into the effect of potassium. Excess potassium in the extracellular fluid causes heart muscles to become dilated and slowing heart rate. So excess of potassium traveling in the blood and the extracellular space will cause the heart to become dilated and slows down the heart rate. Large potassium concentration decreases the membrane potential of the muscles, and this in turn decreases the intensity of action potential and cardiac muscle contraction strength. So what does this mean? So we know that the ionic gradient across the muscles is very crucial for proper action potentials to occur. And one of these, the ionic gradient, is the potassium ion gradient, which is way higher in the intracellular environment with respect to the extracellular environment. Large potassium concentration increase in the extracellular environment disrupts This ionic potassium ionic gradient, and thus the action potential or ability of the cardiac muscle to undergo action potential is hampered. And hence we see a decrease in cardiac muscle contraction. Next, we look into the effect of calcium ions. Excess calcium ions causes the heart to enter a spasmodic contraction. That is, the heart contracts and does not relax at all. This occurs, thus the large calcium ions concentration forcibly initiate and maintain the cardiac contractile process. Maintain the cardiac contraction process. Why? And this is also very intuitive because, as we have seen multiple times, muscles need calcium ion in order to contract, and large amounts of calcium in the extracellular space allows large amount of calcium ions to get into the cells during an action potential. Once these excess calcium ions get into the cell, they bind to the troponin tropomyosin complex and allow the muscles to contract. But after action potential is initiated and dies down again, the calcium needs to be pumped out of the intracellular environment. But in case of a large concentration of calcium ions in the extracellular space, this pumping is not effective and calcium ions accumulate in the intracellular environment. And hence, the next action potential comes before enough calcium ions have been pumped out of the intracellular environment, which results in further and further accumulation of calcium ions in the intracellular environment till the point that the calcium ions continuously initiate and maintain the cardiac contractile process. The heart remains in the state of contraction and cannot relax. So this is obviously very dangerous. And low calcium ion concentration causes similar effect to high potassium concentration. And obviously we know the high potassium concentration, as we have just seen, dilates and slows down the heart rate. And it is obvious that low calcium concentration will do the same, because calcium ions, as I just said, is necessary for contraction of muscles. Low calcium means less contraction, and obviously that will also cause less strength of contraction and lowering of heart rate. Finally, we look into the effect of temperature. Increased body temperature causes greatly increased heart rate, sometimes to as fast as double the normal amount. Decreased temperature causes a greatly decreased heart rate, falling to as low as a few beats per minute. This second condition is known as hypothermia. These effects presumably result from the fact that heat increases the permeability of cardiac muscle membrane to the ions that control the heart rate. So potassium, calcium, sodium, the permeability of the cardiac muscles to these ions increases. And thus this result in an acceleration of the self-excitation process that we will look into just in a moment. But you can just imagine it as that the membrane gets more permeable to the ions, so they can easily cross the cell membrane and initiate the heartbeat faster, and thus the overall heart rate is accelerated. Next, we look into the rhythmical excitation of the heart. So the heart has two special properties, which is first of which is that it can generate rhythmical electrical impulses to cause the contraction of the heart. So the rate of the heart beating is something that is dictated by the heart itself. So there are mechanisms in the heart present that dictate the rate of heartbeat. No external stimulation is required to start the heart to beat. So even if you see say culture of cells or culture of cardiac cells in a petri plate, you will see after some particular stage they start beating. There is no necessity of an external agent to initiate the beating of the heart. Further, the heart has special mechanism for conducting impulses rapidly throughout it. So once this rhythmical electrical impulse is generated, it is spontaneously delivered to other parts or transmitted to other parts of the heart. This is done by specialized excitatory and conductive system of the heart. The first of these, this system is the sinus node, that also known as the SA node or the sinoatrial node, which is present right here. So at the top of the atrium, the normal rhythmical impulse. So this is the sinoatrial node, which is present at the top of the atria right here. This node is very important and is absolutely necessary for generating the normal rhythmical impulses. So this node is responsible for dictating the rate of heartbeat and is also known as the pacemaker of the heart. The next part of the excitatory conductive system is the AV node, which is present right here. The AV node, the impulses travel from the sinus node to the AV node, but they are delayed at the AV node, the necessity of which we will look into in further slides. After which the impulse travels from the AV node to the through the AV bundle and penetrates into the ventricles. We have to understand that the muscle layer in between the atria and the ventricles is completely non-conductive. So the impulses can only pass from the atria to the ventricle via the AV node and the AV bundle. Next, after passing the AV bundle, the impulses are distributed throughout the cardiac muscles on the ventral side via special Purkinje fibers, which are muscle fibers, but their main function is to transmit electrical impulses. All of these are specialized muscle fibers whose main function is not to contract but transmit electrical impulses throughout the heart. So next we will look into how exactly the sinus nodal rhythmicity is generated. The sinus node is a small, flattened, ellipsoidal strip of specialized cardiac muscle, which is located in the superior posterolateral wall of the right atrium, immediately below and slightly lateral to the opening of the superior vena cava. So this was a lot to digest, and we'll look into it a little by little. So we go back to the previous picture, and we have to understand that ordinarily location on an organ or in location in our body is represented by three separate axes, which is the superior superior inferior axis, which runs like this. So we can imagine the superior, for simplicity, you can imagine the superior inferior axis to run like this, top to bottom. Dorsal ventral axis, which is from the back to the front, and the mediolateral axis, which is from the middle to the distant most part of the particular organ. So the location of the sinus node, if given by this particular system, will be superior, like on the top part, posterior lateral, which means posterior means towards our back, and lateral means towards the edge of the heart, not towards the middle. And thus the location of the sinus node is given as superior posterolateral, located on the posterolateral wall of the right atrium, immediately below and slightly lateral to the opening of the superior vena cava. Superior vena cava is one of the great veins that carry blood into the right atrium, so it is located slightly lateral. So once we have the location and the structure of the sinus node in place, let us look into the function of it. The sinus node fibers have a resting potential of minus 55 millivolts, which is way less than the ordinary minus 85 millivolts of the cardiac muscle. But these are specially capable of self-excitation. That is, they can generate spontaneous action potential. So they can generate action potential within themselves without any external help. So as we have seen in the previous lecture, Cardiac muscles have two types of protein channels, which are the normal fast sodium channels and the slow calcium channels. At minus 55 millivolts resting potential, the sodium channels are already inactivated, and thus any action potential that is generated in the sinus node is generated by the slow calcium channels alone. Thus, the atrial node action potential is slower to develop due to it being initiated by the calcium channels, and also slow to return to the resting potential. As we can see right here, it takes a long time for the sinus node to start from its hyperpolarized point to reach the threshold at which action potential will start. Ordinarily, as we can see in the example of the ventricular muscle, this shooting up of the membrane potential from the resting potential value to the threshold is very fast. But in case of the sinus node, especially as the calcium channels are very slow, this takes a long time. This takes a long time. Thus, this is one of the properties of the sinus node itself. So, how does it affect the generation of the rhythmicity of the heartbeat as well as the self-excitation? Between heartbeats, influx of positively charged sodium ion causes slow rise in resting membrane potential in the positive direction. So, the first thing we have to understand about the sinus node is that it has a lot of leaky sodium channels or cationic channels. So, if you go back and remember, we have discussed leaky channels before, which allow free movement of cations through them. So, sodium ions can easily move through them without along their concentration gradient without any hindrance. So, these are essentially open gates via which sodium can enter the cell. And as we know, the concentration of sodium ion in the extracellular fluid is way higher than the intracellular fluid, and sodium just walks in through the open leaky channels into the cell. And as this occurs, the membrane potential of the cardiac muscle decreases. So, the membrane potential of the sinus node is already at minus 55 millivolts. As the sodium or positive charge starts entering the cardiac muscle, this membrane potential further increases to minus 50, minus 40. And when the potential reaches the threshold voltage of about minus 40 millivolts, the voltage activated sodium calcium channels are now open. And this opening of voltage activated sodium calcium channels allow a huge amount of sodium and calcium to enter into the cardiac muscles, and this generates the action potential. So to summarize, this flow portion is the one in which the leaky channels are allowing sodium ions to enter till a threshold is reached, and at this point voltage gated channels open, allow a large amount of sodium and calcium to enter the cell, and thus action potential occurs. Therefore, basically the inherent leakiness of the sinus node fibers to sodium and calcium ions cause their self-excitation. We have to keep in mind that all of this is happening only in the sinus node itself, and the inherent leakiness of the sinus node fibers is what causes their self-excitation. But what causes it to return back to its original resting potential? The calcium channels become inactivated by themselves within 150 milliseconds after opening, which is a much longer time if you remember the fraction of a second it took to inactivate in case of skeletal muscles, but they do inactivate. And second, a greatly increased number of potassium channels also. So potassium, as we remember, the intracellular concentration of potassium is higher than the extracellular concentration. This greatly increased number of potassium channels allows the potassium ions to exit the cell. As they are exiting, they are essentially taking positive ions out, and thus a movement of positive ions out of the cell allows the membrane potential to again progress back to the negative levels, which is the membrane is back. Membrane now begins its journey towards repolarization. The potassium channels remain open for a few tenths of a second, hyperpolarizing the membrane to -55 to -60 millivolts. So the potassium channels remain open for longer than required and hyperpolarize the cell below the resting potential. This new state of hyperpolarization is not maintained forever, because in the next few tenths of a second, the potassium channels close and the inward leakage of sodium and calcium ions through the leaky channels begin all over again, which then restore the membrane potential towards the resting value, and then again start increasing the membrane potential towards the -40 millivolt threshold for the next action potential to occur. This is repeated throughout a person's life and is what dictates the rate of heartbeat. So once the internodal transmission of the once the cardiac impulse is generated in the sinus node, how does it travel to various parts of the heart muscle? The action potential spreads through the entire atrial muscle mass from the SA node and eventually to the AV. So from the SA node present somewhere here, the action potential travels to the AV node. The velocity at which the impulse travels to the atrial muscle, the wall of the atrial muscle, is 0.3 millimeter per second, and through the atrial fibers, which are connected directly to the AV node, is about 1 meter per second. So obviously, before the impulse travels through the atrial muscles to the AV node, the atrial fibers bring the impulse directly to the AV. The impulse is delayed at the AV node before passing to the ventricles. A total of 0.16 second delay is generated before the impulse reaches to the contracting muscles of the ventricle. So AV node delays the impulse before it allows its passage through the AV bundle. And into the ventricular site. Once the impulse reaches the ventricles, it is carried by special Purkinje fibers, through which impulses travel at a very fast rate, 1.5 to 4 meter per second. So these allow a fast transmission of the impulse towards the ventricular muscle, The left and right Purkinje fibers carry the impulse to the left and right ventricular muscle. And finally, the transmission of action potential through the ventricular muscle fibers themselves is again slow, around 0.3 to 0.5 meter per second. So this is the whole transmission of the cardiac impulse as occurs from the sinus node to the furthest parts of the ventricular muscles. Next, we have a schematic showing the transmission of cardiac impulse through the heart with the time of appearance of cardiac impulse. So the cardiac impulse is generated at the SA node and it travels through the atrial fibers, obviously around the closest to the SA node. The impulse reaches simultaneously, but by the time it reaches the AV node, it is already delayed by about 0.03 seconds. We see that the other parts of the atrial muscles are conductive, and the impulse has already reached some of the other parts of the atrial muscle. Once at the AV node, it takes a long time to pass through the AV bundle, and a time delay of 0.16 seconds is generated. After which, the Purkinje fibers takes this impulse to the cardiac muscles in the ventricles at a very fast rate. So a whole delay of around 0.03 seconds, a whole delay of 0.13 seconds is generated in the AV node, but the finally Purkinje fibers take the whole impulse to the cardiac muscles within another 0.03 seconds. And thus, finally, the impulse reaches the ventricular muscles and they can contract. There is an obvious reason for this AV node and the AV bundle to delay the impulse. The reason is that as we have just seen in the previous lecture, the atria are primer pumps. That is, they prime the ventricles to pump more efficiently. This happens by this can only happen. The priming of the ventricle, that is flow of blood to the ventricle from the atria, can only happen if the atria contract before the ventricle. So the delay allows the atria to already contract, push the blood into the ventricles, and once after the delay the impulse reaches the ventricular muscles and they contract, they have already collected most of the blood from the atria. The excess 20% that we have already discussed. So the atria essentially contribute excess 20% of blood to the ventricles, and in order to do that they have to beat they have to contract a bit faster than the ventricles do, and that delay between the atrial contraction and ventricular contraction is generated at the AV node and the AV bundle. So we move on to the control of excitation and conduction in the heart. As I said before, the sinus node is the pacemaker of the heart, or the tissue that dictates the rate of heartbeat. The sinus node drives the heart rate by controlling the rate at which the cardiac muscles depolarize and contract. But other fibers also have a rhythmical excitation property. The AV nodes also can generate rhythmical excitation or self-excitation at a rate of 40 to 60 times per minute. And Purkinje fibers also can generate this, can also generate the self-excitation at a rate of 15 to 40 times per minute. But the sinus node actually has a self-excitation rate of 72 times per minute, and thus before these muscles can self-excite by themselves, the sinus node already depolarizes them, and thus is the one that controls the rate at which the heart will pump. But occasionally, some other part of the heart develops a rhythmical discharge rate that is more rapid than that of the sinus node. So we have to keep in mind that even if there are many self-excitatory fibers present all over the heart, the one that self-excites the fastest or at the greatest rate is the one that will dictate the rate of heart beating. In normal conditions, the 72 beats per minute of the sinus node is what dictates the heartbeat, and that is why the sinus node is known as the pacemaker of the heart. But in case due to some abnormality, if the AV node or the Purkinje fibers will start beating faster than the sinus node, then the pacemaker of the heart shifts to that to the AV node or the excited Purkinje fibers, and they now dictate the rate at which the heart will beat. So in this case, the AV node or the excited Purkinje fibers are known as the ectopic pacemakers of the heart. Or ectopic, the word ectopic means is something occurs in a place it should not. And as we have seen, the pacemaker of the heart is ordinarily the sinus node, and thus if the pacemaker shifts to the AV node and the excited Purkinje fibers, they are now known as the ectopic pacemaker of the heart. So moving on, we have already discussed the control of cardiac rhythm by the nervous system, but now we will look into it in greater detail. To start off, we will look at stimulation by the parasympathetic nervous system. Parasympathetic nervous system stimulates the atrial part of the heart via the vagus nerve. The acetylcholine released at vagal nerve endings greatly increases the permeability of the fiber membranes to potassium ions, which allows rapid leakage of potassium out of the conducting fibers. As we have repeatedly seen that the potassium ion concentration is way greater intracellularly than extracellularly. The permeability increase to potassium ions will cause most potassium ions to start to exit the cardiac fibers. As potassium ions are exiting the cardiac fibers, there is positive charge moving out of the fibers, and thus there is an increased negativity inside the fibers themselves, an effect which is known as hyperpolarization. which makes these excitable tissues much less excitable. In the sinus node itself, the state of hyperpolarization decreases the resting membrane potential of the sinus nodal fibers to a level considerably more negative than usual. So, we have seen that the resting membrane potential of sinus node is around minus 55 millivolts. As more and more potassium ions start moving out of the sinus node, the resting membrane potential of the sinus node starts to drop to minus 70 to minus 80 millivolt, a level that is considerably more negative than the normal nodes. And as we have seen in the action potential curve of sinus node, there is a really slow building up period till the resting membrane potential reaches the minus 40 threshold, after which the action potential shoots up. In case in case the resting membrane potential falls even lower, so the graph will start at a point like this, and the rise of the rise of the membrane potential via the leaky sodium and potassium, sodium and calcium channels will take even longer till the minus 40 threshold is reached and sinus node can undergo an action potential. And thus, this parasympathetic stimulation lowers the rate of heart. Therefore, initial rise in the sinus nodal membrane potential caused by inward sodium and calcium leakage requires much longer to reach the threshold potential for excitation. This greatly slows down the rate of rhythmicity of these nodal fibers. So this slowing down of the rhythmicity can occur to a great rate, where strong parasympathetic stimulation can even stop the beating of the heart. But such an instance is temporary. Once the sinus node stops beating, we have seen that there are other excitatory tissues such as the AV node and the Purkinje fibers. So once the sinus node has stopped being the pacemaker of the heart, it normally shifts, as we have just discussed, to the AV node. And AV node now starts directing the beating of the heart and acts as the pacemaker. And thus the AV node now allows the heart to restart beating at 40 to 60 beats per minute, keeping the heart beating and not stopping it, as that will be obviously lethal. So we move on to effect of sympathetic stimulation on cardiac rhythm and conduction. Stimulation of sympathetic nerves releases the hormone norepinephrine at the sympathetic nerve endings. In sinus node, an increase in sodium calcium permeability causes a more positive resting potential and also causes increased rate of upward drift of diastolic membrane potential towards threshold level for self-excitation. So once sympathetic nerves release norepinephrine, this causes an opposite effect in the sinus. What this causes is to increase the permeability of sodium and calcium in the cardiac fibers. Once the permeability of sodium and calcium increases, sodium and calcium rush into the fibers themselves and start decreasing the resting membrane potential from a minus 55 millivolt resting potential, and it starts increases to minus 50, minus 45. As we have just discussed, once the threshold of minus 40 is reached, that is the only time the sinus node can undergo a action potential. But as the resting membrane potential is increasing little by little towards this threshold, the time required for the sinus node to cross the threshold decreases, and in turn the heart rate increases. Thus, this is what is written here, that an increased rate of upward drift of diastolic membrane potential, so the increase in the resting membrane potential of the sinus node towards the threshold level for self-excitation, accelerates the self-excitation process and therefore increases heart rate. In the AV node and the AV bundle, increased sodium calcium permeability makes it easier for the action potential to excite them, and each succeeding portion of the conducting fiber bundles are excited much faster than normal, which decreases the conduction time from atria to ventricles. So the delay that we were seeing, about 0.16 seconds, that decreases to a lower amount, which also plays a role in increases the overall high rate of heartbeat. So that was all I had today. In this particular lecture, we looked into how exactly the rhythmicity of the heart, is maintained, as well as how it is regulated by the intrinsic as well as the intrinsic mechanism known as the Frank-Starling mechanism, as well as the nervous system itself. I thank you for listening to this video.